Hello everyone, welcome to another video from DNA Bioscience. We have been talking about the immunology lectures and today's topic is precipitation reactions. So in school days we all have learned that the precipitation means when you add some solid material to a solution. Suppose we take a test tube and then add a solution, then add a solid material to it. Then after some time you get the result, that is the precipitation occurs. That means a solid insoluble mass is settled down in the lower part of the test tube. So that is called the precipitate and the reaction is called the precipitation. So this is the normal definition of a precipitation for a layman. So let's discuss the precipitation reactions in immunological terms. Reactions between the antigen and antibody. So you can see in this picture, suppose this is a test tube. You have poured the antibodies and you have also poured the antigens. So the antigens are soluble in nature and the antibodies are also soluble in nature, and they form a zone of equivalence that is called the visible precipitate, which is insoluble in nature. So, what is the precipitation reaction? So, precipitation reaction is an interaction between antigen and an antibody where the antibodies are allowed to react with the antigens. Both the antigens and antibodies diffuse towards each other, and at the point in which they meet, that is the optimal proportion, they will form a visible precipitate, that is called the zone of equivalence. So the precipitation reaction is of three types. First one is the radial immunodiffusion. So always in biology try to break the words. First is radial. Radial means something which deals about the radius. Suppose in this case this part is called the radius or the thickness. The otherwise you can say the thickness portion or the radial portion. The immuno means we have already learnt about the immuno word in the type 3 hypersensitivity reaction. Immuno means the complex found between the antigens and antibodies. And diffusion, we know that there is a movement. So the radial immunodiffusion, it is a type of single diffusion, but it occurs in the double dimension. It is used to obtain an quantitative level of an antigen. So what is the motive of this experiment? The radial immunodiffusion, the main motive is to obtain the quantitative level of an antigen. So you can find that what concentration of antigen is present in a sample. So it is a quantitative method. What do we need to do? Suppose this is a slide or a plate. So we need to pour the agar with the antibodies. So in the plate now, the agar with the antibodies, they spread in a homogeneous manner. Homogeneous means in the same concentration. In the plate, the antibodies are placed homogeneously. Then you mark the wells. This is the first well, this is the second well, this is third, and this is fourth. So we mark 4 wells, and in that well we need to pour the antigen solution. Okay, agar with the antibody, they are incorporated into a plate, and then the whales are created, and the antigens are placed in holes punched into the agar. So as the antigen diffuses into the gel, this is the antigen, the yellow part. As the antigen diffuses into the gel, it reacts with the antibody. So in the surrounding, antibodies are present. We have already discussed that. So inside the well, the antigen is present. So the antigen starts diffusing and it reacts with the antibody. And after some time, it forms a equivalence point where it forms a precipitation ring. So the diameter of the ring is directly proportional to the concentration of the antigen. Since the antibody concentration, we have already discussed that it is constant and it is homogeneous throughout the plate. So by running different concentration of a standard antigen in this direction, the antigen concentration decreases. So in the first well, the antigen concentration is more as compared to the second, as compared to the third and fourth. So by running different concentrations of a standard antigen, one can draw a standard curve from which one can calculate the or quantify the amount of antigen present in a unknown sample. So this is all about the radial immunodiffusion. This is the plate, or you can say this is the slide. You pour the agar media, so that becomes the agar plate. Then you pour the antibody into it, then punctuate and make some wells. Then pour the antigens of different concentration in the wells. In first well, the antigen concentration is more as compared to second. After some time, the antigen starts diffusing towards the antibody. As in the surrounding, the antibodies are present. It forms a precipitation ring or the equivalence point, which diameter is directly proportional to the antigen concentration. So in this case, the precipitation ring is quite smaller than the first one, as the antigen concentration is less than the first one, and in this case the concentration is least. So this is all about the precipitation and the single diffusion precipitation, that is called the radial immunodiffusion or the Mancini method. And the antibody which cause the precipitation reaction is called the precipitin. Here the antibodies are called precipitin, and the antigens that induce the formation of specific precipitin, that is called precipitinogen. So this is all about the precipitation reaction and the radial immunodiffusion. In the next class we will be discussing about the rest of the part of the precipitation reaction. I hope it's helpful. Please subscribe to my channel to get more such videos, and if you like the video, please hit the like buttons and please share your comments. Thank you.